Okay, welcome everyone to our 42nd Students of Phrag webinar. My name is Ned Willig and I am with the Great Lakes Commission, and I'm hosting the webinar today on behalf of the Great Lakes Phragmites Collaborative. Our presenter today is Emily Tarsa from Utah State University, and she will be presenting her work on seed-based wetland restoration following Phragmites australis removal and harnessing seed traits and systems modeling to promote native plant establishment. And Emily is the second of two speakers we've had from Utah State University. So we're really excited to hear a little bit more about what's going on in the Great Salt Lake wetlands out west. So with that being said, I'm really excited to introduce today's speaker, Emily Tarsa from the Utah State University. Emily is a fifth-year PhD candidate in wetland ecology and in the Wetland Ecology and Restoration Lab at Utah State University. Her interests include understanding the competition interactions between native and invasive plants, seed ecology, and restoration ecology of wetlands. Prior to starting her PhD program, Emily worked in the restoration field where she oversaw several wetland restoration projects and was involved in many education and outreach efforts to share the importance of wetlands with the communities. Emily, thank you so much for sharing your work with us today. I will now transfer presentation control over to you and let you take it away.
Awesome. Thanks, Ned. Let me know if you're able to see that.
Yeah, it looks great. So take it away.
Awesome. So thanks everyone for being here today. And thanks for the introduction, Ned. So today I'm going to talk a little bit about a piece of my dissertation work focused on seed-based wetland restoration following Phragmites australis removal. And I'm specifically going to talk about this approach that we're using to improve native plant recovery using functional seed traits and systems modeling. And I just want to preface this talk by mentioning that a lot of the analysis and results from this research are still underway. But I'm excited to share with you some of what we've been focusing on, some of our preliminary results, and kind of the near future vision for this research.
Hey, Emily, I think you went on mute accidentally, so we are having trouble hearing you.
Can you hear me?
Yes, you can hear you now.
Sorry about that.
No problem.
Let's see if I can get back to my speaker. Let's see. Okay, so introducing our study system. So we work in Great Salt Lake wetlands, which, if you aren't familiar, it's a large wetland complex in the Intermountain West region, which is a pretty arid region generally. It's really significant because it provides a lot of breeding ground and food resources for migratory bird species especially. So you can see on this map here, this comes from the Audubon Society. So this is just the western portion of the United States. All of these lines represent different pathways of migratory bird movement throughout this region, and the Great Salt Lake is located right here. So you can see it's kind of a central hub of movement because it falls right along the Central and Pacific flyways. So it's really significant for migratory bird populations. And then there's also a lot of regional significance. So Utah itself is a very arid state, and 85% of the wetlands are located along the Great Salt Lake. So there's a number of really important ecosystem functions and services that are provided by the system on a regional scale. So like the Great Lakes and many other wetlands in North America, the Great Salt Lake has been really heavily invaded by Phragmites australis. And this was a relatively recent invasion. So in the late 80s, there were some really destructive flooding events on Great Salt Lake wetlands. And as those floods receded, there was a lot of disturbed, unvegetated area that was exposed, which was just the perfect opportunity for invasion by Phragmites. And so it was found in Great Salt Lake, or at least in the herbarium record, first in 1993, and it has since covered over 23,000 acres. Obviously really problematic in this region because it's very aggressive, as we all know. It outcompetes a lot of those native plant species. So it really threatens the habitat quality and the food resources for migratory birds and other waterfowl species. And Phragmites is also problematic because it spreads very rapidly. So we see spread through above ground stolons, underground rhizomes, and also a lot of spread by seed. So over the past decade, our lab has done a lot of research on effective control techniques and treatment for Phragmites. This work was carried out by Chad Craney and Christine Rohal, pictured here. And they did a 5 to 6 year experiment looking at the different timing of herbicide applications, different types of herbicide, and then different timing of mowing treatments. And they found that a summer mow plus a fall glyphosate was really effective at killing Phragmites. It reduced seed heads and it reduced the seeds in the seed bank, but that should be done for at least 3 years. And so that's kind of the currently applied control technique on Great Salt Lake wetlands to knock back Phragmites. And if you're interested in hearing more about Chad and Christine's research, this is our lab website right here. They have a nice extension document that summarizes a lot of their findings. So that's a great reference if you want to check that out. Unfortunately, in these areas that they treated and in a lot of areas that are treated by managers on Great Salt Lake, we see that native plant recovery is really limited after Phragmites treatment. So there's either no native plants coming back, or the native plants that we see coming back aren't those desirable habitat forming graminoid species that we would hope to see. So because we aren't seeing these desirable native species returning on their own, active revegetation has been a big focus of our research efforts and then also management efforts on Great Salt Lake. And so there's a number of different active revegetation strategies, but in theory, seed-based restoration is a really promising restoration strategy because it's less expensive, less labor intensive to install, especially when you're working over large areas, which is kind of the scale of the problem on Great Salt Lake. There's hundreds and hundreds of acres that need to be restored. And kind of this added benefit of using seeds in restoration is it allows you to increase the adaptive potential of your plant community. So because it's more feasible to collect seeds from many different source populations, you end up with seed mixes that have more genotypic diversity. And then with more genotypic diversity, there's a better chance that some of those genotypes will do well in one type of condition, whereas other genotypes might do well in a different type of condition. So you're kind of hedging your bets that you'll have native plant establishment across a wide range of conditions. And this is also a really nice way to promote climate resiliency. So when we're thinking about what these systems might look like with climate change projections in the future, using this adaptive potential strategy with seeds is a nice way to prepare these sites for what those projected future changes might be. And so seed-based restoration has a lot of advantages. Unfortunately, seeding outcomes are really unpredictable and they're largely unsuccessful. And this is especially true in wetlands because we see a lot of these challenging conditions that can be conducive to seedling mortality. So wetlands are really hydrologically dynamic, so it could be water levels could be too high or too low and can cause mortality. Wetlands are also really prone to invasive species establishment. There can be a lot of propagule pressure, so there's a lot of competition that's happening at these early life stages. And then generally these wetlands are really degraded and disturbed in some cases. So generally the environmental conditions may not be as conducive to seedling establishment. I want to just take a step back and talk about these early life stages where we see a lot of this mortality because seeds and seedlings are very, very vulnerable. And so we have these kind of 4 distinct life stages that a plant goes through in the early portion of its life cycle. So initially we have, the seed germination. So you have this radicle that's protruding from the seed. Emergence is when the above ground shoot kind of breaks through the soil, and you get some root formation. Establishment occurs when that little seedling starts to photosynthesize on its own versus relying on the reserves in the seeds themselves. And then you just have this end of season survival. And I'm going to overlay this graph. So this was a study done in upland systems where they sowed a known number of seeds, and then they tracked how many of those seeds germinated, how many emerged, established, and survived. And you can see there's this huge mortality event that occurs between germination and emergence. And overall, in a lot of these species, we see about 5 to 10% of the seeds that we initially sowed survived at the end of the season. So this was done in upland systems where a lot of this research is occurring. It's kind of a new research frontier in wetlands, but we have seen some of these same trends. And if you tuned into Ray's presentation last week, she showed some graphs as well where we see kind of the same pattern of mortality in a lot of our species that we work with. So this obviously isn't good for restoration. If you don't have native plants establishing, there's this opportunity for reinvasion, and it's also just a waste of seed and a waste of money. And so the big question then becomes, how do we overcome this bottleneck in recovery or in recruitment to maximize native plant establishment after seeding? And to answer this question, my research is focused on understanding the mechanisms that are driving these early life stages so we can understand what's happening and improve conditions or improve species selection to maximize native plant recovery when we seed. So I'm taking a functional trait approach, and I just want to take a step back. Whoops. Just take a step back and talk about functional traits themselves, what they are, why they're important. So functional traits are just morphological or physiological characteristics that impact plant growth, reproduction, and survival. And here's just some examples of some adult plant functional traits. So we have things like plant height, root length and structure, parenchyma, which is really important in wetland species, leaf mass per area, a number of seeds produced. So these are just some examples of traits that adult plants might have. And these are really important because they allow us to predict where we might expect plants to be distributed in a system. It can also help us predict how plants might interact with each other and compete with each other. And functional traits themselves are a really interesting, I guess, topic to study because it's kind of like a common currency between plants. So if you look at the functional traits of a plant, you can compare those functional traits and kind of generalize across a wide range of species instead of just focusing on species-specific characteristics. So there's been a lot of research on adult plant functional traits, less so on these functional traits that drive regeneration. However, there are functional traits driving these stages from germination to emergence, establishment, and seedling survival. And that's the focus of a lot of my research. So I want to talk about some examples of what these traits are using pictures from our study specifically. So there are some of these traits that are inherent to the seed, so they don't necessarily change across environmental conditions. And here's just a few examples. So we studied things like seed dimensions, which tells us how long seeds might persist in the soil seed bank. We also looked at seed coat thickness. So for our study species, we took cross sections and measured the thickness of the seed coat at a few different locations. And this tells us again how long seeds might last in the seed bank, but can also tell us things about how quickly seeds might germinate. Then we also looked at seed mass. So seed mass is very commonly studied. We know that generally larger seeds have better seedling survival, especially in really harsh conditions. And so all of these pictures here are seeds from the same species. And you can see there's quite a bit of variation in both seed dimensions and seed mass. We also looked at a few functional traits. We also looked at a few functional traits at the seedling stage. So these are traits that vary across environmental conditions. Oh no, sorry guys. Okay, hopefully it stays there this time. So these vary across environmental conditions. And so some of the traits that we looked at here were germination traits, like time to germination, so how quickly it took for seeds to germinate. Relative growth rate, so how fast the above ground shoot grew in these different environmental conditions, and then root elongation rate, so how quickly the roots elongate in different environmental conditions. And overall, understanding these functional traits can help us make more targeted restoration decisions. So this isn't just a science for the sake of science question, but there are real practical applications for using these traits to help guide restoration through seed. And so some of those decisions that we can make with this functional trait knowledge is which species we might want to seed. So comparing traits across these species can help us predict which species will likely outcompete Phrag or will establish more reliably from seed. So we can understand which species might germinate faster or have growth rates or have faster root elongation rates. And here's just a photo that I want to share that kind of depicts this point. So here are the 7 focal species of our study, including Phragmites australis. These seeds were grown in ideal environmental conditions, just moist soil at optimum temperatures. And we harvested the seed just a day after germination for each of these species and just captured photos just to kind of see what was going on. And you can see even just one day post germination, there's a ton of variation across species. So saltgrass specifically grows quite rapidly and germinates pretty quickly. So we already see some tall shoot formation. The roots already elongating quite a bit, even faster than Phragmites itself. And then some of our other perennial graminoid species, like our Bulrush species, you can see that germination is occurring at a much slower rate. So this might tell us that saltgrass could be a very good competitor at these early stages by looking at this trait. It can also help us better target where we might want to source or collect our seeds from. So within a species, we would expect to see a lot of variation in regeneration traits across source populations because there's different environmental conditions at each site and there's different selective pressures. And so here's just another picture example that I want to show. So these are Alkali bulrush plants. We collected seeds in this study at many different locations, which we'll talk about in a minute. But here we can see Alkali bulrush growing at Clear Lake waterfowl management area, which is obviously very dry, compared to the Alkali bulrush that we collected from Warm Springs waterfowl management area, which is a really wet, inundated wetland. And so we might expect here that seed sources that experience drier, hotter conditions could have better survival in dry, hot conditions. So that's another way that we can kind of think about preparing for future projected conditions at our site. Okay, so jumping into kind of the methods, a really quick overview. So again, we looked at the variation in these traits between species. So here are our 7 species that we worked with, including Phragmites. And then we're also really interested in how much variation there is within species to kind of inform seed source selection. So for these species, we collected seeds at many different wetlands across the Intermountain West. So all the way up north in these Montana wetlands, some collected down in Idaho. We collected from wetlands in Great Salt Lake and other Utah wetlands, and then down south in Nevada. Okay, and here is a really quick rundown of the methods. So after we, after we collected all these seeds, we brought them back to the lab. We took seed trait measurements. So we looked at those things like seed dimension, seed coat thickness, seed mass. We also looked at seed buoyancy, which is kind of an interesting way to capture how these seeds are dispersing across the landscape. After we collected these seed trait measurements, we conducted this 1.5-year-long growth chamber experiment So we had a number of different growth chambers operating under 3 different temperature regimes. So we did a cool, a warm, a hot temperature, and we had 2 different moisture levels, so a saturated condition and a dry condition, and we manipulated that with polyethylene glycol. And so for each of those temperature and moisture level regimes, we sowed every species times source population combination, and we tracked individual germination. And then several days after germination, we harvested the above ground and below ground parts of these seedlings. We scanned images to measure the length and the surface area of the roots and the shoots, and then we took fresh and dry weight of the roots and shoots, and those are kind of the basic components of calculating those really important growth metrics like relative growth rate and root elongation rate. And then we just wrapped up a final greenhouse experiment related to all this research. So we took those same environmental conditions, those same species and sources, and we grew them out in a greenhouse setting. And in each of these containers, so they're each a species and source combination, we tracked end of season % cover. And then on this other side of the containers, we also tracked the probability of seeds transitioning across those life stages. So every day we came in and we marked, a germinated seed, and then we tracked it throughout its life stages, or we measured if it ended up dying during the course of the experiment, just to get the probability of transition across life stages. And so, as I mentioned at the beginning of the presentation, we're still in the really early stages of data analysis, but we do have a few initial findings. So I want to just highlight a couple of those findings and then talk about where we're going. So this is just looking at some germination metrics that we've seen. So we found that there is quite a lot of variation across species, which isn't too unexpected. And in this top graph, this is looking at germination proportion. So it's the proportion of seeds that germinated out of all of the seeds that we planted. We have our different temperature treatments here. So our cool, warm, hot, and then we have our wet and dry conditions. And then all of these colored dots represent our different species. So you can see there's quite a lot of variation in germination proportion. Saltgrass in particular generally had the highest proportion of germinating seeds, followed by our Juncus species and Phragmites. And then Alkali bulrush consistently had really low germination proportion across environmental conditions. Surprisingly, we didn't see a decrease in germination in our dry conditions. So that's a bit surprising, and we're still kind of piecing through the data to understand why that may have occurred. Part of our thought is that we may not see those trends until slightly later. So when we start looking at seedling traits, for example, we might see some more differences between these wet and dry treatments. And then this bottom graph is looking at Germination Synchrony, which is another really interesting germination metric, and it's an index that tells us how close together all of these seeds germinate. So if there's a synchrony index of 1, that means all of the seeds germinated on the same day. If it's an index of 0, it means that germination occurred, or it's more spread out across many days. So this is really interesting because it can tell us how these seeds and seedlings may compete with each other. You would expect that if you have all of your seeds germinating really rapidly and very reliably, then those plants kind of get a head start and can preempt and acquire more resources before other plants have a chance to germinate and establish. So again, saltgrass had much, much higher synchrony in its germination pattern compared to Phragmites and many of these other native species that we tested. So from these germination traits, just kind of these initial results, we might expect that saltgrass is a fairly tough competitor in the germination and establishment phase because the seeds germinate more quickly and at a higher rate than Phragmites or even the other native species. And that is something that we've seen borne out in other field experiments as well. Like saltgrass is generally pretty good at establishing. So we also found a lot of variation within a species, and here's just one example graph. This is looking at the variation in seed mass for Alkali bulrush, which is this plant down here on the bottom right corner. So in this graph on the y-axis, we have the seed mass of an individual seed. And then on our x-axis, we have our different seed sources. So these are the different areas we collected seeds. And they're ordered from our southernmost population. So they're ordered from the southernmost population to the northernmost population. And we can see that in general, as we increase in latitude, we get increasing alkali bulrush seed mass that is pretty significant. And again, seed mass generally has implications for higher seedling survival in harsh conditions. So we talked about, the variation that we've seen both between and within species. Again, we're kind of working through a lot of this data, but we are planning to use a systems model to predict kind of the restoration outcomes based on these different seed traits. And this is just a schematic of the model itself. We have in the green here, these are our different life stages. And so in the greenhouse, we tracked the probability of transition across germination, emergence, seedling establishment, and end of season survival. And so we also measured these seed traits and seedling traits and are going to correlate those to their how they influence the probability of transition across these life stages, and then how these environmental conditions, temperature and water availability, influence these seedling traits that then change the probability of transition. And kind of our final output here is percent cover, which we measured in the greenhouse and is obviously a metric that managers in the field care about. So this is just roughly what our model is looking like right now, and we're still in the process of kind of parameterizing the model, but our overall vision is to use kind of this web-based interface so managers can go in and kind of plug in their different scenarios and get these percent cover expectations and suggestions on what species or sources they should use based on their their restoration site and kind of their context. So there could be some different ways of looking at it. Managers might input their budget in the restoration area, and it will output which species and sources they should use and kind of the ideal environmental conditions to maximize native cover. Alternatively, it could be that managers input their desired species and water availability at the site, and then it tells them what seed sources they should use, when they should seed, which relates to temperature, in order to maximize native cover. And so the model itself is really adaptive and iterative. In our very long-term vision, after we kind of reparameterize this initial model, is incorporating new native species with different functional groups, incorporating more environmental conditions and spatially explicit recommendations, and having a better way to account for competition. But the nice thing about this model is because it's based in functional traits, it can be applied widely beyond just Great Salt Lake wetlands themselves. And I do want to just, before I end, just talk about this resource that we've put together. So we have kind of consolidated all of the results of Ray's research, some of my research, and some of the other research that we've been doing in our lab, focused on seed-based restoration. And we've developed this University of Utah extension document. And so it's just a nice document that summarizes all of our findings and provides guidance on how to choose the ideal revegetation site, where you should source seeds, how to select species, and all of those kind of important considerations when you're restoring wetlands via seed. This is currently in review, but I was talking to Ned earlier, and I can send this around to him when it is fully published, which should be in the next month or two. And then I also included my email here if you wanted to reach out to me directly about this or anything else that I've talked about today. And then before I close, I just want to thank all of the lab technicians and graduate students, both current and former, that have been involved in this research. It's been a pretty big undertaking over the years. So I've had an awesome crew of people assisting in this process. A lot of thanks to my committee members for guiding this research. And then over on the right-hand side, you can see all the different funding agencies that have had a big impact on the work that we've been able to do. With that, I will take any questions.
Awesome. Thank you so much, Emily. That was really great. Really fascinating work that you have going on. So again, if anyone has any questions, feel free to put them in the chat box. I think the first question I had for you, Emily, is just a clarifying on the experiment that you were running there. When you had the seeds germinating, were those all kind of like one species in a box, or did you have a mix of species kind of in together in those different scenarios?
Yeah, that's a great clarifying question. So in this experiment, we had one species and source population in a box. So we didn't get a chance to look at competition in this experiment, but definitely future iterations of this work will include how those species and sources directly compete with other species.
And would the future research also think about just like addressing Phragmites, how the species, a combination of species might work together to have kind of a net benefit of different, like a bulrush versus the saltgrass being all together, and whether mixes are better to compete against Phragmites?
Yeah, absolutely. Yeah. So a lot of my research is focused on like very specific and individual mechanisms. But there is a lot of research in our lab. So Ray has done some of this, and then we have another graduate student, Karina Hebert, who is looking at what it's like, or what the outcomes are when you mix a bunch of native species and species that are annuals versus perennial species, and how that might help combat Phragmites reinvasion.
Okay, great. One additional question on the resource you mentioned. I was hoping you could talk a little bit more about that and whether that is a specific to the Great Salt Lake context, or if that's information that can be applied both in the Great Lakes and wetlands kind of across the US and Canada.
Yeah, that's a good question. So the resource itself was written with Great Salt Lake, in collaboration with Great Salt Lake managers, talks about a lot of the research that we do, which is mainly specific to Great Salt Lake. But a lot of those concepts are pretty generalizable. So which species you should choose, you know, just thinking about different considerations that are beyond just specific species to our systems, but different functional groups based on your different restoration goals. So there's definitely pieces of it that can be applied to a broader wetland context, but a lot of it is focused on Great Salt Lake.
Okay. Gotcha. Is there anyone else on the call that has any questions? I guess one more question about the kind of the different functional traits, and how did it looks like you were looking at seed mass because of survivability with additional mass, and then also synchrony. Were there other ones that you kind of considered or found were more important or less important based on literature or work done in your lab? Or how did you kind of choose those two metrics to focus on?
Yeah, so there's so this area of research is kind of an emerging research frontier. So there is some of those traits like seed mass and root elongation rate that has pretty direct links to seedling survival, but a lot of these traits are still kind of underexplored. So we decided to choose kind of a wide range of traits that we hypothesized might have some influence on seedling survival. And then in our modeling process, we'll be able to kind of piece apart which traits matter and which traits don't. So that's definitely, yeah, stay tuned for kind of the results on that end.
Mm-hmm. And I saw with your resource, there was we have one question coming in here. So it says it sounds like the tool providing suggestions for seed coverage and mix is applicable, but beyond study sites. In what regions will seed species be assessed for wider application?
Yeah, so obviously our focus again is on Great Salt Lake, but because the model and that online tool based on the model itself is based on functional traits, it will be applicable to other species that are in similar functional groups as the species that we found. So right now in those restoration scenarios, it kind of outputs very specific species like hardstem bulrush from Clear Lake waterfowl management area. But future iterations of that model could include things that are like, well, you're looking for species in this functional group that may exhibit these traits, and they're expected to perform better and reach this level of percent cover. So it can definitely be, used in a wider application beyond this region.
I guess following up with the research on that, has there been thought for the Salt Lake context on an iterative learning model, just as research comes back, and if you have managers using the outputs from that model to kind of put that back in as a like iterative learning and adaptive management type framework?
Yeah, absolutely. That is definitely the intent for this model. And so in this initial model, we've worked really closely with a lot of managers doing restoration on the Great Salt Lake to kind of make sure we're identifying the things that they are looking for when they're restoring a site. And the intention in the future with future graduate students is to actually apply, our expected predictions from this model in a field setting and just see how well it performs in the field, and then kind of go back and revise the model over, I'm sure, many iterations to kind of improve and refine model predictions.
Gotcha. And just curious, kind of one more question about the model too. You've done a lot of really interesting research with the seeds as they're growing, but I saw there was the kind of budget question. I know that's a big issue with the cost of restoration. Is that something you were involved with the estimating the costs of going around to get all these seeds, or is that something different where there's another resource for that, understanding the kind of costs related with all of the different seed selection?
Yeah, it's interesting. So the seeds that we collected for this research, we harvested on our own. But we work really closely with a lot of the seed suppliers. So we have a pretty good estimate of what it costs to actually, purchase those seeds and have them sourced. So that's kind of where we're planning to pull in that budget aspect. And then we do have another graduate student in our lab starting in the fall who is focused specifically on just the cost effectiveness. So different planting methods, their exact cost, and kind of looking at the output. So some of that data I think can be pulled into the model. But for right now, we're just using these estimates from our seed source suppliers.
Okay, great. Yeah, that's interesting stuff. Are there any other questions for Emily? I think this has been really interesting research. I think I might go ahead and just share my screen again. And again, yeah, thank you, Emily, so much for sharing your research, and for those on the call and those following the webinar kind of after the fact with the recording, we will be following up with the resources that were shared here as well, and try to feature those on the Phragmites website, the Great Lakes Phragmites website. So as I mentioned, the webinar recordings are available on the greatlakesphragmites.net/resources/webinars, and you can be able to view this webinar as well as previous recordings through that. For those on the webinar, our next webinar will be June 8th with Sylvia Jacobson sharing research based in Maryland I think at the Smithsonian Environmental Research Center on wetland recovery following Phragmites removal. And you can register for that on our site as well. So want to thank Emily again. This was really great research, and also thanking the U.S. Geological Survey for their support to provide this presentation. We are thankful for all the support we get and for that. And this will conclude the webinar. So if you have any questions or comments, feel free to contact us at phragmites@glc.org, and we will follow up and be sharing resources shortly. So thank you again, everyone, and have a great day.